I'd always wanted to be a scientist, and when I was in college and graduate school, I actually went to graduate school in theoretical physics, and I was just very attracted to the computational sciences, also mathematical modeling. But in my first year of graduate school, actually quite early, I realized I wanted to do something that was more directly helping people. And after that, I decided, biology is what I wanted to do. I was really excited. It was around the time that the human genome was announced as having been sequenced. So then I started a postdoc in computational biology, and I've been working in this field ever since. How can we help patients live longer? How can we cure their cancer so it doesn't come back? It's difficult for a number of reasons. And one of the big challenges is that cancers, even if we treat them with therapies we have now, which tend to work, the cancer comes back. And we don't really understand why in a lot of cases. One of the aspects that's the most important is that the immune system will actually attack most cancers, but it varies in how well it does. So what we're really interested in my lab is figuring out ways to improve that ability of immune cells to really completely get rid of cancers.
The way we develop images for pathology might change over time. We might use different equipment, different procedures, staining procedure, et cetera. And therefore, being able to have flexible models that do not overfit and that can be accurate across different data sets is vitally important for accurate diagnoses.
Tissue AI is the field that I work in, which is the study of tissues, your muscles, your liver, your lungs, your brain, et cetera, all these different tissues, and how to understand what's going on in them, then develop treatments. AI, artificial intelligence, this is the analysis of data. And the reason that this is kind of a big area right now is because for the most part, even though people know what tissues are, most of our treatments that we study in biology and also at the clinical level, a lot of them are not really designed around the behaviors of the tissue themselves. One of the exciting things that we can do now is that we can profile tissues in ways that are really leaps and bounds beyond what we could do in the past, which is we can now measure almost every gene in a cell and its behavior across the entire scope of a tissue. So for example, within the human genome, we have roughly 20,000 genes, and I can now tell you what is the expression of every gene in every cell and where that is localized within the tissue. We can also do similar things for proteins. Generally, it's harder to do as many proteins, but we can measure those individually. And so we end up with these pictures of tissues in not kind of the dimensions that we normally look at. I think people normally think of looking at a picture, it's like red, green, and blue. You combine those, and then you kind of see in color. But instead, we're now seeing in thousands of colors at once, because we can see the behavior of every gene. And so now we have this challenge of these amazing pictures where we can see either in 2D by looking at a piece of a tumor, or we can, in some cases, see in 3D, where we can take multiple sections of a tumor and kind of stitch them together and make a 3D image. And we can look at the behavior of many, many genes all at once. And so the exciting thing about that is now we have so much more information about not only where the cells are, but also about how all the genes are behaving within each cell. But we need ways of being able to analyze all that data all at once. And that's why people such as myself, computational biologists, it's such an exciting time for us to use these AI approaches to analyze the data.